In this short video, we will address the question, what is flow cytometry? Flow cytometry is a technology used to characterize different cellular populations in a mixture of cells. This technology is often used in the diagnosis and classification of hematopoietic neoplasms. With flow cytometry, we use the ability of cells to scatter light in order to measure physical properties of a cell, such as cell size or cytoplasmic granularity, and as well, we can use flow cytometry to detect antigens expressed either in or on a cell by staining a mixture of cells with fluorescently labeled antibodies and then looking for expression of the antigens to which they bind. A variety of sample types can be appropriate for flow cytometry, but for clinical flow cytometry, it's important that samples are sent to the laboratory fresh, ideally within 24 hours of collection. If a sample is going to take time to reach the laboratory, it's useful to submit it in a nutrient media, such as rPMI, but it's critical that samples for clinical flow cytometry not be placed in any sort of fixative solution, such as formalin. Flow cytometry does require that cells are in a liquid suspension, so a variety of liquid samples may be appropriate for flow cytometry, and they include things like blood, bone marrow aspirate samples, or a variety of body fluids, things like CSF, ascites fluid, or pleural fluid, for instance. In addition to samples that start as liquid samples, solid samples can be used for flow cytometry if you disaggregate them or chop them up and suspend them in fluid. And using this technique, we can do flow cytometry on solid samples, such as lymph node tissue, or a variety of other solid tissue biopsies. All right, let's talk a little bit now about how flow cytometry works. As I mentioned, flow cytometry can give you a sense of various physical properties of a cell. With flow cytometry, we can get, for instance, a sense of cellular size, and as well, a sense of the complexity of a cell or cytoplasmic granularity of the cell. In order to do this, flow cytometry harnesses the ability of a cell to scatter light. So let's consider an example of how this works. Now in the center of your screen, you have a cell, which is shown in gray, and has various antigens expressed on the surface, some orange and some blue. For flow cytometry, we'll start by shining a light on the cell, typically in the form of a laser. And as that light hits the cell, it's deflected in different